Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. We will now discuss biosynthesis of terpenoids and outline in this lecture number 36. So, let us go to the board. So, before I start talking, so there are three aspects which I am going to cover. One is the an outline of MV and MEP pathways, and then various isopenoids, and then monoterpenoids and introduction. So, let us now go to the what are the terpenoids. So, biosynthesis of terpenoids. So, terpenoids are lipid soluble molecules. So, they are generally not water soluble, and terpenoids comprises of more than 10,000 molecules, and they play important role in the survival of the plant. Now, several or a significant number of terpenoids are important for our different uses. So, one important area is essential oil. So, essential oils are nothing but it is basically a mixture of terpenoid components. So, why we call them essential oil? Because basically they have the essence, and these are the a mixture of terpenoids which we isolate in the form of liquid, and they are volatile. So, volatile terpenoids in liquid form having essence, we can call them essential oil, and when they are present inside the cell, we normally call them as internal pool of volatiles. So, that is in general, we will come to that. So, how we start is basically that we basically talk about the diversity of different terpenoid molecules. As I said in the very beginning of the specialized metabolism class, that all the specialized metabolism groups originate from primary metabolism. Therefore, terpenoids are not come under any exception. So, they are very much the offshoot product of the primary metabolism. So, for example, say the three acetyl CoA. Acetyl CoA, and this side we write glyceraldehyde 3 phosphate plus pyruvate. So, these two joins together and ultimately contributes to the formation of MEP pathway. Whereas, from acetyl CoA, MVA pathway originates. Now, both these pathways contributes towards the formation of isopentenyl pyrophosphate. IPP, which are 5 carbon compounds C5, and this can be converted into dimethyl allyl pyrophosphate, that is also C5. and from here different molecules originate. So, for example, from DMAPP joins with one molecule of IPP, and it forms GPP, geranyl pyrophosphate, and this GPP basically contributes towards the formation of monoterpenes. Monoterpenes are C10. So, GPP is also C10 and DMAPP C5, IPP C5. So, 2 C5 joins and makes C10. DMAPP joins with 2 molecules of IPP and forms FPP, which is farnesyl pyrophosphate. Gap is there. Okay. And this again by different reactions contributes towards the formation of sesquiterpene. So, this is C15. So, sesquiterpene SESQUITERPENES. This is also C15. These are C15 compounds. From FPP, another route moves towards the formation of squalene, SQUALENE, which are C30 compounds, and squalene ultimately contributes towards the formation of triterpenes. C30. Now, DMAPP with 3 molecules of IPP can form GGPP, geranyl pyrophosphate, which can contribute towards the formation of diterpenes. It is C20 compound. GGPP is also C20. And another route, 2 molecules of GGPP joins together and contribute towards the formation of phytoene. Phytoene are also C40 compounds, and phytoene contributes towards the formation of tetraterpenes. Is C40. So, this is in brief the origin of different terpenes from primary metabolites either through MEP or MVA pathway. Well, so now in the next slide, I will briefly talk about the steps of MEP and MVA pathway. So, first in brief, okay. In brief only I will cover, not in that detail. So, that is an outline of MEP pathway. Okay, first I put first I discuss MVA, then I will discuss MEP. So, MVA originates from two molecules of acetyl CoA. This you should remember, and that makes acetoacetyl CoA. And that subsequently makes HMG CoA, and then it will make mevalonic acid. So, from there the MVA now name derives. The MVA stands for mevalonic acid pathway. Then it will be mevalonate phosphate, then mevalonate diphosphate, then isopentenyl diphosphate or isopentenyl pyrophosphate, which we call IPP. So, now let us join this with arrows. So, and the enzymes responsible are AACT, acetoacetyl CoA synthase, and then HMGS, then HMGR, HMG CoA synthase, HMG CoA reductase, then mevalonate kinase, then subsequent other enzymes. We will not go into that, as at the end it forms IPP. So, and isopentenyl diphosphate converted into dimethyl allyl diphosphate, that I will show separately. So, this is in brief the MVA pathway, and MVA pathway is basically operating in the cytosol. That is what you must remember, that this pathway operates in the cytosol. Whereas, now if we talk about the MEP pathway, MEP pathway requires joining of glyceraldehyde 3 phosphate plus pyruvate or pyruvic acid, that leads to the formation of DXP, deoxyxylulose phosphate. Commonly called DXP, and the enzyme is called DXP synthase, and this converted into MEP, methyl erythritol phosphate. Methyl D, okay, let me write the full. Methyl C D, okay, methyl D four phosphate, okay, simply phosphate. This we call MEP. MEP subsequently converted into different products like CDPME, which I am not going to discuss. CDPME, then CDP, MEP, then so on, and finally it comes to HMBPP, and from here it makes IPP P. So, if I join with arrows, this is DXR, and DXP deoxyxylulose phosphate reductase, this is called DXR, this is called MCT, then this is called CMK, this is HDR, this is MCS, HDS, there are so many, but you only try to remember DXP synthase or DXS and DXR, because these are the two important and rate limiting enzyme. Here HMG CoA synthase and HMG CoA reductase, these are the important regulatory enzymes. So, this you must try to remember, and this pathway is basically operating in the plastid, chloroplast. This is the fundamental difference. Now, one more important thing I should tell. So, MEP pathway was discovered in late 90s in the microbial system, and subsequently it was found in the plant system, only in animal system, only in the parasites or protozoa only have the MEP pathway, but not the other animals. And whereas MVA pathway is universal, And one interesting point is this, that there are several inhibitors available to block the MEP pathway. One of this is phosphomydomycin, so one antibiotic. So, that basically inhibits the DXS, and this is used as one of the alternative antimalarial drug, because as I said that MEP pathway operates only in protozoa. So, the Plasmodium, the causal organism is a protozoa. So, giving an inhibitor of MEP pathway to block that is basically you are blocking the metabolism and eventually that will die. So, that is the beauty. Now, how MEP pathway discovered? Okay, MEP pathway discovered from the bacterial point of view, that is a different story, but from the plant point of view, when scientists they were working on the essential oils production in cell cultures. Okay, so, they were not convinced that it is the MVA pathway which is basically modulating the pathway. So, feeding the precursors of MVA pathway did not have any effect, even blocking the MVA pathway did not have much effect. Therefore, from that point of time, a scientist called Charlood, B.V. Charlood from King's College London. So, he was of the opinion that there must be another alternative pathway exist in the plant system which we are not aware of. And much later when MEP pathway was described, so it was found that MEP pathway significantly contributes to this terpene monoterpene biosynthesis, and indeed MEP pathway's contribution is more than MVA, particularly for this monoterpene biosynthesis, which is basically operating inside the plastid. Okay. So, that aspect we will come. So, this is in brief about the MEP and MVA pathway. We will not go into that much detail, as I said, where detailing is required, there we will move. Okay. Now, let me also give you another overview of this MEP pathway. So, what are the different products originate from MEP pathway? That means, what we can write, diverse products originate from MEP pathway. So, MEP pathway is also called DXP pathway also, it is same. So, you can write in bracket, because DXP was the DXP is the first product of the pathway. Okay. And what I said that this operates inside the plastid. Okay. Okay. And if I start with IPP, so from this pathway it produces IPP. IPP is also produced outside the plastid by MVA pathway. So, MVA pathway which operates outside the plastid in the cytosol also contributes to IPP. Now, there will be crosstalk between this IPP and this IPP, that means, when there is a shortage of IPP in the plastid, then IPP from the produced in the cytosol can move inside, and whenever there is a need for more IPP in the cytosol, the IPP produced in the plastid can move out. So, and as I said, along with IPP it also produces DMAPP, dimethyl allyl pyrophosphate. Now, DMAPP contributes to different products such as isoprene, irregular terpene, and terpenes also we can say isoprenes, and also this also contributes towards the formation of cytokinin. Okay. And DMAPP, from DMAPP what we see that GPP is formed, from GPP GGPP is formed. Okay. Now, GPP contributes towards the formation of monoterpenes. I am not happy with this arrow. So, and GGPP contributes towards the formation of carotenoids, and GGPP also contributes towards the formation of phytol, and this phytol takes part in the synthesis of chlorophyll, tocopherol, and phyloquinose. And from GGPP another route originates, leading to the formation of different bioactive diterpenes, including gibberellins. GGPP, from GGPP through another route it produces plastoquinone. So, this is in brief different products or diverse products originate from MEP pathway. And from the MVA pathway the important products are, so anyway because of shortage of space let me put it here. If we put the MVA pathway here, so the MVA pathway makes IPP that into DMAPP that makes GPP. Now, this GPP makes FPP, farnesyl pyrophosphate, and this FPP plays important role in the synthesis of sesquiterpenes and triterpenes. One it goes to sesquiterpenes, another route goes towards the formation of triterpenes. And TPP also contributes for GGPP, and this GGPP also contributes towards the formation of polyterpenes. A good example is rubber. So, this is basically the major role played by MVA pathway. Okay. Now, let us go to the concept of isoprenoids. So, isoprenoids are 5 carbon molecule. So, this is the structure of isopentane, and this is the structure of isoprene. So, if you put it in CCH3 double bond CH2 CH double bond CH2. So, isoprenes can make different configurations. For example, one is called head to tail. This is called head to tail. The other one is called head to head. No. কেটে গেলে মুশকিল হচ্ছে। This is called head to head, and another type which is called head to middle. This is called head to middle. Okay. Now, let me show you the example. So, head to tail, the example of such compound is geraniol. Now, head to head, the example is squalene. It's a bit big molecule. So, here is the point of head to head condensation point, and the example of this is squalene, which are C30 compound, geraniol is C10 compounds. Okay. And the example of head to middle is I can also draw, which is here. So, this is the structure of pyrethrin. Pyrethrin 1. So, this is also, so all these, this is also monoterpene. Pyrethrin 1 is monoterpene, but it comes under the point of irregular monoterpene. So, geraniol is monoterpene, squalene is basically triterpene. So, these are the different ways isoprenoids skeletons are modified, leading to formation of diverse products. One more point is this, that how this GPP, GGPPs are formed. So, these are formed by different reactions, but that I may discuss in the next class. So, with this I end this class. So, let me see what I intend to cover. I intend to cover outline of MVA and MEP pathway, this I have covered. Various isoprenoids I have given, and monoterpene and introduction very briefly, we have not yet started. So, we will talk about that in the next class. So, with this I end this class. Thank you very much.